video on your own if you're interested. The types of viruses that treat cancers are called oncolytic viruses. So it's a lytic virus. It's one that causes the lysis of the cell that it infects. So you can imagine sending a virus in to, instead of make one of your healthy cells sick, if the virus specifically took over a cancer cell and then killed the cancer cell, that would be really cool, right? That's the idea behind using these terrible viruses like measles to kill. In the case of measles, they were targeting melanoma. The video is about something that happened in the Mayo Clinic. They treated 2 patients. One patient had long-term remission of her melanoma. It might have been a myeloma. But the other patient didn't have such good results, and they've had trouble getting it to be reproduced. So this is a couple years old now. So rather than show you this video that may be a one-shot thing, I'll leave it there for you to look at. But know that it's something people are working on. There also is more current research being done on using poliovirus to infect brain cancers. Think about this for a second. Poliovirus, what do we say its tropisms are? What kind of tissues does it like to infect?
Intestinal.
Intestinal, but also neural tissues, right? And so they've been doing some experiments trying to treat people with glioblastomas with poliovirus. Now you ask yourself, wow, that sounds great. How far advanced is this? And I'll tell you that Joe Biden's son died of glioblastoma, and John McCain just died of glioblastoma. And you figure they've got access to the best health care in the country probably, right? So it's not advanced far enough. Does that make sense? Which is why I'm not going to show you a video of it. But I want you to know about oncolytic viruses because, or have heard about it at least once, because it may be that during your career as a healthcare professional, these become a standard of care. And so it's good for you to know this is something that people are working on. We'll also talk about immunotherapies for cancer as well. We've had a hard time curing cancer. It's basically trying to figure out your cells, your sick cells from your healthy ones and target only the sick ones. It's really tough medically to do that. Anyway, I would encourage you to watch the video anyway, but I want you to understand that it's experimental. It hasn't worked for more than really one patient, and they're still trying to figure out ways to do it. And it's not the only kind of cancer that's being targeted this way. In some ways, that glioblastoma may be further along. So I won't show you the video, but I will leave it in the slide set. So I have said maybe once to you, but I will probably say it a few more times, I consider this class to have 3 sort of big hurdles. One of the hurdles is metabolism. So it's understanding glycolysis, Krebs, and electron transport. A lot of biochemistry there. The second hurdle is genetics, understanding how DNA makes RNA, makes protein, understanding mutations, okay? Understanding the process of the ribosome, constructing a protein molecule. That's the second hurdle. And the third hurdle is immunology. And so 2 of the 3 hurdles are in this next exam. And we're going to kind of start today with some of the work that I think students find really challenging. And we don't have to rush because I have plenty of time budgeted for these lectures. But at the same time, I understand these concepts are hard. You guys are kind of a quiet group, which is completely fine. But it doesn't. What it is hard for me to judge sometimes if you're with me or you're lost. I try to do that with quicker questions, but I want to encourage you to raise your hands. Sometimes I have a couple real boisterous people and they kind of get the whole class going. And you guys are all so polite. And I love that about you, but I really do want feedback. So if you're stuck, we have time, raise your hand, let's go over it again. Because it's easier to get you unstuck when you first get confused than after we've gone through a ton of material and you're really lost. Does this make sense? Those of you who sit in the back, I know you're there. And you can ask questions too. I'm ready to pick you over the people in the front because I know why you're sitting in the back. Yes?
It's like, I'm not sure I have it. So I look at the slides and look at the book. We talk about a lot about the glycolysis and we're not on the Krebs cycle and then the electron transport. I was wondering, there's a difference between oxidative phosphorylation and there is this difference between the breaking down of these glucose in the bacteria that we're studying?
So over the next couple lectures, we're going to talk about all those things. So I think you're asking questions that are about the upcoming material. And if you get lost along the way, let me know. Are you asking about differences between what's in the slides and in the book in terms of content, or?
The difference between the electron transport chain and the oxidative phosphorylation.
Yeah, so usually, They are, well, they're usually when phosphorylation is happening in the electron transport chain, we tend to refer to that as oxidative phosphorylation. When phosphorylation is happening in glycolysis or Krebs cycle, we call it substrate level phosphorylation. So, but again, we're going to talk about these things, so it's a great question. You're ahead of yourself too, or ahead of where we are, which is fantastic. But we're not there yet if anybody's panicking over it. All right, I hope I've encouraged you to raise your hand. Like I said, this is one of the big hurdles. If you're stuck, probably you've got really good company and you'll just be a hero for raising your hand, because other people will be, oh, thank God someone asked that. So try to be brave and put your hand up, and I'll try to encourage you as much as I can, because I think that it really helps everyone when even one person's asked a question. All right, so here we go. Microbial metabolism 1. I've got 2 lecture slides that we're going to spread over about 3 days. And the process of glycolysis, Krebs, and transport has a lot to do with getting energy for the cell. So I just want to remind you of some terms here. If I want to get energy out of a molecule, we call that what?
Catabolism. Right. Oh. I don't know.
I was about to say.
I don't know.
If I want to get energy out of a molecule, which is another way to say if I want to produce energy. All right, I see lots of clicks in, so we'll kind of do these quickly here. Oopsie. And you guys are saying metabolism is the process of getting energy out of a molecule, right? And then, I guess I don't have, did I have 2 of these? Nope. Okay. All right, so metabolism, when we say the word metabolism, we're actually referring to both catabolism, which is breaking down of molecules, getting energy out, and anabolism, which is building up of molecules. And so this is just a simple schematic showing that in a cell we have catabolic reactions where we take complex molecules like glucose, sometimes they're even more complex, sometimes they're molecules like starch or fat, and we break those down, we release heat, and we make ATP, usually starting with ADP and inorganic phosphate. And these produce, when you're breaking big molecules down, you produce small molecules, and then of course you also break these down all the way to produce carbon dioxide and water, which is not shown in this schematic. If you take little molecules and you build them up into bigger molecules, that's called anabolism. In this case, you're also releasing heat, but you're using ATP to make these molecules. So it takes energy to build things. In cells, when you break things down, you release energy. All right, and both the catabolism and anabolism, that process is called metabolism together. This is another kind of schematic to try to help you organize your thoughts. Here's our glucose molecule. We can undergo catabolism and break it down into smaller molecules like pyruvate and acetyl-CoA, glyceraldehyde 3-phosphate, and not on this chart are things like carbon dioxide. If you want to break it down all the way, you would break it down to CO2. You can also take glucose molecules, or you can take some of these smaller byproducts of glucose breakdown, and you can build amino acids from them. You can build complex sugars. You can build nucleotides. You can build fatty acids. And so as you go through anabolism, you can create new building blocks from glucose. You can then organize these into larger macromolecules, proteins, in the case of bacteria, peptidoglycan, large molecules of nucleic acids, lipids, complex lipids like we might see in a cell membrane. And then when you put all these big macromolecules together, you can assemble them into a cell. All right, so the process of anabolism means taking nutrients like glucose and really creating new cells out of them through a chemical synthesis process. How are we doing so far? All right. So in order for any of these things to happen in metabolism, both anabolism and catabolism, you need a molecule called an enzyme. Right? We've talked about enzymes a little bit, and you've probably talked about them in chemistry in your other classes. How do enzymes work? Well, we're going to talk a little bit about that. It ends up being more important to us in micro than maybe even in some of your other classes, because we not only need to understand enzymes to get how cells function, but it turns out that bacteria make enzymes to break down antibiotics, and it's one of their strategies for survival against the kinds of molecules we send in to try to destroy them. So they're really important for us. here's our enzyme molecule. Here's a substrate molecule. So the enzyme acts on the substrate. So you can see the substrate fits into the active site of the enzyme. And the enzyme is going to break this substrate into two pieces. And so we call this the products. So far, so good. All right, so you could do this in, almost in elementary school. A little simple drawing. But let's start here, okay? Now, enzymes do all sorts of really complicated things. Sometimes they take two molecules and build them into one molecule, right? This would be the way this is drawn, it's a catabolic process, but if we were going this direction, it would be an anabolic process. You know a molecule is an enzyme when it has the ending ASE. ACE. Anything ACE is an enzyme, right? So an oxoreductase is an enzyme that facilitates oxidative reduction reactions. A transferase transfers things from one thing to another. So maybe it's transferring a methyl group. Maybe it's transferring an OH, but it's a transfer it's a transfer protein, right? It's a transferase. A hydrolase adds water and causes things to break down usually. Lyase removes atoms. Isomerase moves things around. So you're taking the same molecule and just leaving the same number of atoms but isomerizing it. So changing the structure in 3 dimensions. Ligase is a kind of a famous enzyme used in genetics because it joins 2 molecules together, usually in DNA, putting 2 pieces of DNA together, creating a phosphate sugar backbone. So enzymes have an ASE backbone. So all of these are enzymes, even though they have really different names. One thing that I think people tend to think about of enzymes being just protein. And a lot of your enzymes are not just protein. A lot of your enzymes actually require other molecules to be functional. And these other molecules are called cofactors and coenzymes. So if an enzyme needs a non-protein component bound to them for activity, then it needs a cofactor. If the cofactors are inorganic, so like a metal ion or an iron sulfur group, then they're called a cofactor. If they're inorganic, then there's a subset of cofactors called a coenzyme. So things like flavins or chemes, that in case of heme it will also bind iron, but it's an organic molecule, we call it a coenzyme. So all cofactors, cofactors include, I think this tends to be a point of confusion, so I'll just draw this a little bit. So in the subset, so these are cofactors, and then coenzymes are a subset of cofactors. If it's not a coenzyme, then it's something inorganic. Like, for example, a metal ion. Does that make sense? And the cofactors that are proteins, or not proteins, that are organic molecules, they're also cofactors, but they're a subset of cofactors called coenzymes. If that's useful, having that diagram up. Okay. It's almost good enough without the screen, huh? Is the screen better? I was saying, because if you do it this way, you can kind of draw on it. Anyway, I'll have to think about that some other time. All right, so when an enzyme requires a cofactor, then we refer to it in the absence of the cofactor as an apoenzyme. So this is a apoenzyme, and you can see it's the protein part of the enzyme, but it's not fully active yet, because in order to be active, it needs its coenzyme. All right? And the coenzyme will bind to the protein, and together the coenzyme and the apoenzyme make something called a holoenzyme, which is an active protein. So here's our substrate, and it won't bind to the apoenzyme, but when you add the cofactor or coenzyme, then this becomes an active molecule. And why do we care about this? Well, we care about it because it turns out a lot of vitamins function this way. So vitamins are often coenzyme, and some of the vitamins that you're familiar with actually function in glycolysis, which is what we're about to talk about. So vitamin B2, also known as riboflavin, is very important in FAD, which is something we're going to talk about in the Krebs cycle, functions in the Krebs cycle. Niacin nicotinic acid is part of NAD. NADP is actually in photosynthesis, but you don't photosynthesize, you guys, none of you look green. But NAD you definitely use. And another one is folic acid, which is actually very important with coenzyme A, also involved in Krebs cycle. All right, so lots of the enzymes that we're talking about, and we're not going to talk, if you take upper division biochemistry in college or get a biochemistry degree, you'll have to memorize all these enzymes and every step and where the carbons go, but for our class, that's not necessary. But I do want you to be familiar with the idea that this is a role that vitamins play. There's a misconception that people need to take supplements in order to get all their vitamins. The supplement industry is a multi-billion-dollar industry. And if you're getting, a reasonable amount of nutrition, and when I say reasonable, I mean, McDonald's every day pretty much does it. It doesn't have, you just have to not be starving. Most people get the vitamins they need. You just need tiny amounts of this. Pretty much every study that I've ever seen says that taking vitamins as a supplement is not necessary. And in fact, a lot of those studies have shown that some of the vitamins that people have taken in excess actually can cause cancer. So for example, vitamin E. People have a great antioxidant. It turns out that it's been associated more with higher levels of cancer instead of reducing cancer. So everything in moderation. If your wallet is hurting your back and you need less, get some of the dollar bills out of it, then you can buy vitamins. That would be a good use of. Otherwise, vitamins aren't going to do anything for your body. There was just another study that came out about fish oil. You heard about fish oil? It's supposed to be good for cardiovascular health. Some people said it was going to be good for mental health. Got DHA and EPA for little kids developing brains. No evidence. They've done these big meta-analyses now. Don't waste your money on fish oil. Not only that, it's bad for the environment. Yes?
So what about for someone who doesn't eat animal products? Do multivitamins actually help at all?
Not really.
Okay.
So if you don't eat animal products, you're probably getting actually a really nice, these vitamins tend to be in veggies. The tricky thing we worry about with someone who has a vegan diet, for example, is just that you want to make sure you get all the essential amino acids.
Okay.
Because I think there's something like 11 amino acids, I can never remember the exact number, that your body can't make from glucose, that it has to get in its diet. And those you need to get from your diet. So if you're eating strictly one kind of plant protein, you might be deficient in an amino acid. But few people who are vegan only eat one kind of plant protein. Usually they eat several different kinds of plants and several different kinds of beans and tofu, and at some point you're getting all the amino acids you need. It's very easy to eat a full balanced diet being vegan. You might start craving steak at some point.
What about iron though?
So that's actually a good point. Heme iron,
Yeah.
the iron that's found in animal products is more bioavailable than iron found in spinach, for example. So keeping track of your iron and potentially supplementing iron might be something that you would want to do. But that would be, you can also take too much iron that you can get toxic. Iron has warning labels on it. If you have little kids, you can actually poison them with eating iron. So you want to take iron as you need it. Menstruating women, because they're losing blood each month, tend to have lower iron levels in their blood than someone who's a man who's not menstruating, or a menopausal woman, or child before puberty. So that's a time when women tend to be kind of anemic. On the other hand, usually people don't, a little bit of a lower iron maybe isn't as big a problem unless you've really lost a lot of blood. Your body usually recovers from it.
So overall, should supplements essentially be prescribed then if you really don't need them?
It'll never happen politically because there are both Democrats and Republicans that are been lobbying by, the multi-billion-dollar industry. I think vitamins alone is $4 billion, but when you add in all the other supplements that are out there, it's like $20, $30 billion a year. And they spend lots of money lobbying Congress to not be regulated at all. So for example, when you buy a prescription drug from the pharmacy, that has to be tested, prove to the FDA that it is what it is and that the amount is what they say. But when you buy a vitamin, those are self-reporting. They've done studies where, I used to show this video to my human biology students, where PBS went and they had just things like echinacea and St. John's wort, and there's one for hot flashes that I can't remember. Anyway, they went and took 4 or 5 of these supplements, these herbal supplements, and they DNA sequenced what was in the bottle. They went to Whole Foods and a bunch of places that you would think would have really high quality products. And in like half of them, there wasn't a trace of the plant that was on the bottle. And in another portion of them, what was in there was actually just grass. And when I say grass, I don't mean the good kind. I mean like the kind you cut on your lawn. You know, so they're not regulated. No one's testing them. So the answer is that those people that tell you at GNC that you need that stuff to be healthy, like I said, if your back hurts, wall's too heavy, that's the only reason I can think of. My back isn't that much pain.
So if your doctor is saying that you need a supplement, like, so in my case, I don't get enough B12 for some reason, I just never do, super
Yeah.
low. They have me take like the 1,000,
Yeah.
something like that, of B12. Does that mean
Folic acid.
I'm not getting it?
Yeah. So no, not necessarily. I think the B vitamins and folic acid and vitamin C, those things are a lot less likely to be risky in terms of the amounts than something like, you know, echinacea or St. John's wort. Those are so partly because they've just been around longer and they're mass produced and everything else. But the answer is they are not regulated. So to the extent that your doctor can prescribe something that's FDA approved, that's great. You know, for a long time, people were taking vitamin C as I don't know, vitamin D is another one where people have been testing, doctors have been testing to see what your vitamin D levels are. The evidence is a little sketchy for that. So I take calcium because I have some osteoporosis in my spine. It runs in my family. And you have to take it with vitamin D because it turns out for years and years we were telling people to take calcium and your body can't really absorb it without vitamin D. So that's just in my lifetime, that recommendation's really changed. And I read a study recently where they were saying, yeah, it's probably not helping that much.
They say after 22
Your bones start to lose calcium, yeah. But I don't necessarily think it helps all that much. What helps your bones keep calcium is estrogen. So the evidence now is that after menopause, it's really good right after menopause or during perimenopause to stay on estrogen to help prevent bone loss. But figure that you're losing bone as you get older. Unfortunately, I've already lost a lot, but you guys are on your way too. And one of the best ways to prevent bone loss or to reduce bone loss is weight-bearing exercise. And so I run. And I run for 2 reasons, and neither one of them is because I like doing it. Actually, I run for 3. It does put me in a better mood. So if I'm cranky, it's a good thing to do. But actually, for the weight-bearing exercise, because osteoporosis runs in my family. And the second reason is because all the vascular flow of blood through the brain seems to reduce the risk of Alzheimer's disease, which also runs in my family. So when I really have trouble getting motivated, I think about, you know, breaking bones and Alzheimer's disease, and I think, okay, I'll go. All right. All right, so we had a little detour on vitamins, but it's kind of a fun, interesting detour. Any questions on this? All right. All right, so let's talk a little bit more about enzymes. Enzymes don't work for free, just like everything else in this world, right? And so enzymes require energy in order to do their jobs. And for any chemical reaction to occur, there must be enough energy in the form of activation energy for the reaction to happen. And in order for the reaction to happen, the enzymes and the substrates have to be in the right orientation. In some cases, that means the substrates have to be oriented properly with respect to each other. And so I just want to talk a little bit about activation energy. Does this look familiar from chemistry? An activation energy diagram? Yeah. So I see lots of people nodding. So lots of times you have reactants that are at higher energy levels than products, but they don't just spontaneously form products. So for example, if I had a beaker full of glucose sitting on the desk, it wouldn't spontaneously form CO2 and water today, or tomorrow, or next year, right? And the reason is because the activation energy, you have to put so much energy into it before you'd be able to break apart those bonds and then form products. With me so far? So what do enzymes do? Well, we could certainly cause my beaker of glucose to form CO2 and water if I stuck a Bunsen burner under it and we incinerated it. So we're going to put energy in, And that's going to cause lots of chemical vibration, and eventually those bonds will break and will form CO2 and water as we burn it. Are your bodies as hot as Bunsen burners?
No.
They're not. And yet, your cells right now are converting a ton of glucose into CO2 and water. And they're doing it without a lot of heat. Nifty, right? So you were thinking that no glucose was being converted to CO2 and water, but I'm just telling you right now that every time you exhale, lots of CO2 is coming out that you just converted from glucose that was in your blood. Okay? It's happening in here, it's just that your bodies are doing it, and they're doing it at 98.6 Fahrenheit or 37 Celsius. How does that work? Well, it's because your body is using enzymes to do it, and so the activation has been, energy has been lowered because the conversion to CO2 and water is happening in the presence of an enzyme. So how does the enzyme reduce the amount of energy? And they do it a couple of different ways, and really I'm just going to talk about two ways, but it's a lot more complicated than what I'm saying, but I'm going to give you a little flavor. One of the ways that enzymes can help a chemical reaction happen is something called induced fit. We used to talk about enzymes when I was in college even, when we talked about enzymes being like a glove on a hand, that the hand was like the substrate and it fit perfectly inside the glove of the enzyme. We actually know that's not actually the best analogy now, because we've done lots of X-ray crystallography studies where we've looked at enzymes in 3 dimensions and looked at how they fit with their substrates. And it turns out that in many cases it's more of an induced fit. So here's our enzyme, this is sucrase, which breaks down sucrose, a glucose and a fructose. It breaks them down. And so you see the sucrose molecule is fitting into the enzyme, but you see how it's kind of being bent? So the enzyme's sort of forcing in 3-dimensionally to be in an awkward, uncomfortable state. And it's actually increasing the energy that the molecule has so that when water comes in, it's easier for a water molecule to break this bond. And the enzyme, of course, is facilitating all this, and it's releasing the 2 products, glucose and fructose. So this is called an induced fit model. It's sort of like a poorly fitting glove that helps cause the substrate, in this case, to break apart. So far, so good? That's induced fit. In many cases, and what I don't have a picture of, and I don't want you to stress about, but if you're interested in it, in many cases, and it may be true for this enzyme, that the substrate actually bonds temporarily. Hydrogen bonds, van der Waals forces, sometimes on the temporary covalent bond with the enzyme in the process of breaking the molecule apart or putting 2 substrate molecules together as a product. So the interactions of enzymes and their substrates to cause chemical reactions to occur can be quite complex. And then, of course, water is coming in and participating as well. Another really important thing that enzymes can do is put substrates in the correct orientation to react. So here's an example of a chemical reaction with no enzyme. And so we have looks like nitrous oxide and chlorine gas. And these are going to react if they hit each other with enough force and they hit each other in the right orientation. So notice in part B here that when the oxygen bounces onto the chlorine, they just bounce off again and no reaction occurs, right? But if the nitrogen bounces onto the chlorine, then we'll form a chemical bond and we'll have a chemical reaction. Are you seeing the difference here? Mm-hmm. So the same amount of energy went into the reaction, but the collision orientation was really important. Now, without an enzyme, this is a random process, right? But when you have an enzyme, the enzyme can put the substrates in the proper orientation. So here you see these 2 amino acids, and they're being fitted together in the grooves of an enzyme to put them together to form a peptide bond perfectly. So enzymes can actually say, you're going to fit this way, like they put the puzzle pieces together in the right orientation so a chemical bond can be formed. Working for you? So things like induced fit and substrate orientation are things that enzymes do so that you don't have to be boiling hot on an incinerator to get your cells to be able to break down glucose and get energy out of it. All right, and so this is just one of the study guide questions. It says, describe a mechanism by which enzymes make chemical reactions happen more quickly and with less activation energy required. And so you could talk here about collision orientation being perfect every time with an enzyme. You could talk here about the induced fit model. And you could really find probably 20 more examples of ways that enzymes work by interacting with their substrate, by facilitating the a water molecule coming in and hydrolyzing the substrate. So there's a lot of ways to answer this that would be correct, but I've given you two that are really straightforward. The induced fit model and being in the right collision orientation. All right, so we just kind of wanted to spend a moment talking about why enzymes are special. Enzymes are considered specific. Now, some enzymes are highly specific, meaning they will only react with one substrate molecule. It has to be exactly that molecule. And some enzymes are a little bit more generic, so they might interact with a category of molecules. This is sort of showing the idea here that here's your protein, your enzyme, and here's a little tiny substrate, and it's fitting nicely inside the active site. And in fact, notice that the enzyme itself changes shape when the substrate is present, okay? Usually interacting with chemical bonds with the substrate and facilitating the change that it's about to cause to happen. All right. No discussion of enzymes would be complete without talking about the kinds of things that damage enzymes. We've already talked about mesophiles, and we said that mesophiles like to be at about 37 degrees. That's where they have their maximum activity, which is what this is showing here. And that most pathogens are mesophiles, so they basically like to be at your body temperature. When an organism adapts to be at body temperature of a person, then what happens is its enzymes are going to function optimally at the temperature of the person. So this graph shows you temperature on this axis and enzymatic activity on this axis. And what you see is that as the temperature goes up, the enzymatic activity increases, but then if the temperature keeps going up, suddenly it dies off. Do you remember in chemistry learning that for every, I think it's 10 degrees, a reaction doubles the rate? Okay, so what happens in biology is that that rule works fine until you denature your enzyme. So as you get above 50 degrees, the heat that's present causes a lot of kinetic energy in the system to start putting pressure or force on those bonds that are holding the protein together. So remember we talked about protein structure. We said it had primary structure and secondary structure, tertiary structure. In the case of multi-peptide proteins, it has quaternary structure. And we said that the protein is held together by forces, so it's got potentially disulfide bridges. It's got van der Waals forces, it's hydrogen bonding. It might have ionic bridges, positive and negatively charged amino acids attracted to each other. All those are holding the protein into its active three-dimensional shape. As you heat it, the molecule starts to vibrate, the kinetic energy gets hotter and hotter, and eventually it starts to disrupt those interactions. And when the protein starts to unfold, then you go from this active enzyme to an inactive enzyme.